Well, thanks everyone for coming to this session. This session's topic is Dissecting Emerging Modalities and Mechanism for Treating CNS Disease. My name is Charlie Yang. I'm a VP of Equity Research at Bank of America, and to my left is Professor Ole, Director of Neurodegeneration Research Institute at McLean Hospital. And he is the expert in the AI and brain health, so I'll let him start with the introduction.
Yeah, thank you, Charlie. We have a very exciting panel for you today, and I just want to reflect on a second that actually the Medical Innovation Forum 2023 is about CNS brain health, oncology, and immunology and inflammation. So although we're going to speak about brain degeneration, we're also going to try to venture about the future, perhaps, of some of these treatments. But the innovation gap is present. We have new modalities, and of course, we're always looking for mechanisms of treating these devastating diseases, as you've heard in the previous panels. So we're going to go through in the order here, first with Lisa, and then Kasper and Rob, to hear from them what they and their companies are working on right now. Lisa.
Thank you. Lisa Ricciardi. I'm the president and CEO of Cognition Therapeutics. We're working on Alzheimer's disease, dementia with Lewy bodies, and also dry age-related macular degeneration, targeting the sigma-2 receptor, which if you follow the whole amyloid cascade, we work at displacing oligomers. It's about right in the middle of that cascade before you get to the plaque that everyone's been peeling out. And we have targeted this receptor because of its ability to address cellular regulation, functions like autophagy, cholesterol handling, vesicle trafficking, things of this nature, the kinds of things that fall apart under stress as we age and lead to neurodegenerative diseases. So pleasure to be here. Thank you.
Hi, I'm Kasper Roet, CEO and co-founder of QurAlis, a company that I co-founded together with two Harvard professors, actually, Kevin Egan and Clifford Wolf, to develop precision therapies for ALS. It was very nice to hear Governor Healy's comments on all the inventions that were made here. She missed one that brought me to Boston. It was actually the ability to create human neuronal model systems from patients. I think that really is transforming the field. And it is really what also created the programs at QurAlis to use motor neurons from patients to be able to identify potential targets for disease treatments and to test potential therapeutics. It led to the discovery of a potassium channel called Kv7.2, which is a precision medicine program that we're bringing forward for sporadic ALS patients and is in the clinic now, healthy volunteer study. And also the discovery of the missplicing of stathmin-2, a microtubule stabilizer, a protein that is essential for the innervation of the muscles, for the integrity of the neuromuscular innervation. Both targets are misspliced, and also the discovery of missplicing as a key driver of disease etiology in ALS comes from these stem cell models. And so I'm very excited to be here today and to talk about emerging technologies.
Good afternoon, everyone. I'm Robert Scanaden, the Chief Scientific Officer at Verge Genomics. Verge is a company that sits at the intersection of tech and biotech. We continue with Kasper's comments in it with an all-in-human platform where we utilize human multi-omic data that's analyzed in our AI-enabled platform to derive new targets for therapeutic intervention in neurodegenerative and other types of diseases. We've used our platform to discover a new target in ALS called PIK5, which is involved in endolysosomal dysfunction, which is a common pathologic feature of ALS and other neurodegenerative diseases. And we've advanced the small molecule inhibitor of this kinase into clinical development.
Great. So thank you very much for those introductions. So our panel is about innovation gaps. So what are we lacking? What are we missing to establish new modalities, perhaps, as tools in treatments? So what would you think here on the panel? What are the new technologies in this space? And maybe, Kasper, you can start, because I know you've been working on stem cells, on antisense, and various other modalities. So what do you think are the sort of emerging new techs that you trust?
Well, I think it's a very exciting time at the moment. We have small molecules, antibodies, antisense oligonucleotides, siRNAs, gene therapy, AAVs, and other viruses, and then also CRISPR genome editing technologies. But when we look at the diseases that we want to treat, the patients that we want to help, and the targets within the cells that we want to target, that actually limits our ability to use many of these modalities. And so this is, for instance, the reason why at QurAlis we use small molecules and antisense oligonucleotides, because they can either penetrate the blood-brain barrier relatively easily, or we can inject them directly into the spinal cord, have good distribution through spinal cord tissue and brain tissue, such as we have with the antisense oligonucleotide, and we can correct it to correct splicing and basically bring proteins back. So at this moment, I honestly feel like we've accomplished so much in modality development that we can now choose to go after a specific target with a modality that is appropriate, and I think that's the right approach to help as many patients as we can.
Thank you. And so Rob, I mean, as you mentioned, the company you're working at now starts almost like a genomic platform, but you're also using computing power, right, to do computing modeling. How do you see these new technologies develop, and what are the gaps that you need to fill, you believe, in our field of CNS disease?
Thanks, Ole, for the question. I do believe that the major gaps that we still face in the field is really about translating our preclinical research into the clinic. We've addressed that through a number of different mechanisms. We heavily lean on iPSC technologies to really study human pathological processes in human disease-relevant cells. And I think this has become fundamental in the field by understanding how different therapeutic approaches can intersect with disease-relevant processes. I think this gets around a lot of the translational hurdles that the field has faced traditionally using other types of, say, heterologous cell models or animal models to really drive target discovery and therapeutics evaluation. I think by adopting these human cell model approaches in a modality agnostic approach, we can really make fundamental advances towards investigating the processes of these diseases.
Thank you. I think that's a very helpful answer. So Lisa, you have experience. So would you call yourself modality agnostic, or how do you approach the problem from target to drug?
Well, so being a business executive and not a scientist like my colleagues here on this dais, I can say I walked into a company and inherited a small molecule program. The founding scientist, a chemist and a neurologist together said, let's back into what is the mechanism, what's the target that's going to allow us to protect synapses over the course of Alzheimer's disease. And through years of work, summed up in about 30 seconds, they identified the sigma-2 receptor and its regulation of cellular activity. So what we have are small molecules. And quite honestly, when I think about it, Ole, from the perspective of with monoclonals, what % of drug gets across to the blood through the blood-brain barrier versus what we know about our program, which is in excess of a ratio of 1, I think those are some of the things that we're making easier with this approach. Modality agnostic, I think you're right. I happen to be in an environment where we're very clearly investing more in more small molecules. The other thing is we have work going on in the back of the eye. Okay, everyone has heard about intravitreal injections for 20 years, right? Macugen, 2 weeks ago, Iveric getting acquired by Takeda, all these things. And I don't think patients over the last 20 years have gotten to like these injections anymore. We have an oral drug. Now, couldn't that be a game changer for patients? And frankly, for all the healthcare costs associated with it, to get that oral modality to treat a condition in the back of the eye.
Yeah, thank you. So Charlie, you had a more global question, right?
Right. So actually, Professor Ole, given your experience and your work in the brain health and AI field, and I know you've been on the board for several companies as well, what's your perspective here, when you think about the emerging innovations in this space? How would you look at it, perhaps even at a higher level?
Yeah, to give the other panelists a little bit of a break here. Thank you for the question. Well, I feel a bit responsible. We're talking about brain health, but just what Rob said, we're now dealing with human data, which is very different than when we started, say, 20 years ago, even in neurobiology. And I've had the fortune over the last couple of years to work with AI companies and huge assemblies of multi-omic human data. So not just genetic, but actually patient data, say from Alzheimer's disease, which includes imaging modalities. So PET scans, we heard from Chris Wiebacher. We have data on metabolomics. And what we can do with the AI models, which is what I've never seen before, is to link then all the patient demographics through massive modeling, through Bayesian, I mean, it's basically a probability what connects to what influences what, to these various 44 different outcomes related to dementia. And what is so exciting, I think, for brain health in the future, like we did for heart disease, is that we see things we've never seen before. So I think AI in that sense for brain health and as a new modality. So the platforms we've used, as Rob mentioned, as genomic platforms. Now we have a new layer, which is massive amounts of human data. And I think that modeling will also give us maybe new diagnostics. And like for heart disease, you know, we go from breaking up amyloid beta to synaptic health, as Lisa says. And I'm very excited about thinking of AI as a modality, actually.
Charlie, I can't help but ask the audience, the Framingham study. Am I the only one here who remembers when that was the biggest deal? Do you remember this, Framingham? It's not far from here, the Framingham study, longitudinal. I believe it was nurses in the beginning, and we thought, oh my God, so much data to mine. And then I listened to Ole and I go, wow. Lucky those of us working on neuro conditions, because hopefully the speed of progress will be so much quicker because the tools are better. Good quality data, but the kinds of tools you're talking about should allow us to make a lot more advances than the humble beginnings of the Framingham study. Or in Scandinavia, in the Alzheimer's disease, we're talking about the finger trials, similar kinds of community-based data collection, but what you're talking about is another order of insight.
Yeah, and I think, you know, you understand the investment we heard from previous panels. We're talking about tens of billions of dollars investment, right? And, you know, the promise of precision medicine. But I think we're going to see it happen without anybody perhaps noticing. And so genomic data helps, you know, in ALS. I mean, we have drugs, right? You work in QurAlis for SOD, you can comment on. But I think in the future, too, you can pull out these data sets and then inform the clinical trial and also inform, you know, outcomes and healthcare and so on. So maybe Kasper, I don't know, both of you probably have comments about this.
I just, for me, this touches on the core of ALS, which is the TDP-43 biology. TDP-43 is a protein in the nucleus that regulates the expression of other genes. When it moves out of the nucleus, those genes dysregulate, basically. And we found that dysregulation is really at the heart of the disease. It's present in over 90% of ALS patients. And this is what we're targeting with QurAlis. We're targeting to restore back these proteins, Kv7, the gene is KCNQ2, STAthmin2, super important for cell health and for cell stability. But TDP-43 is a stress protein. And so it basically tells you that when the cell is stressed, it turns into a pathological state. And normally in a healthy neuron, that stress protein would just go out for a little bit and go back in. In disease, it goes out and it doesn't come back in. And it leads to a very dangerous process and in the end to the demise of neurons. Now, a very exciting new discovery in the field, and this is only a couple of months old, is that we can detect this protein moving out of the nucleus in neurons in the plasma of patients before they get sick. And so now we have a stress protein that is basically the hallmark of disease and really the driver of all these disease processes that we can potentially find in people before they are sick. Now, of course, like many other diseases, ALS is largely a sporadic disease, and it's very hard at this moment to envision screening a population to find you before you get sick. But in the end, maybe that's possible, and at that moment, we can maybe prevent and keep the brain healthy before it turns into a pathological state. So from a futuristic perspective, I'm very excited about that. At this moment, we have to be concrete, and we have to bring therapies to patients that we know are going to have an impact right now.
We're going to follow with you, Rob. I just want to say, I think that what you said there is very exciting, because as you heard from the previous panels, many of the diseases we see, of protein aggregate diseases, are actually not addressing synaptic health. You heard it from Lisa, right? So you can have a massive load, you can be 95 years old, and have as much amyloid as the worst patients, but your brain may actually work very well. So this type of brain health that you mentioned, that you can pick up a signal from a cell that's starting to degenerate, I think could be extremely worthwhile. So Rob, I'm sure you have a comment on this.
Yeah, absolutely. Thanks, Ole. I think to Ole's earlier point, our ability to integrate more complex, different levels of human data, genetics, transcriptomics, metabolomics, looking at lipidomics, is really going to be able to help us understand and interrogate these pathological processes. And the precision medicine aspect of this really become to the forefront when we're able to integrate that with clinical metadata, understand what patients manifest what types of disease, and find the right therapeutic approaches for treating those patients.
Yeah. Right. So let's say that that was sort of our perspective on how we move the field forward, right? But then in drug development, and many of you have been part of this, and I actually have as well, much of it is in the sort of the devil is in the detail idea. So even to the point that when you take a drug to the FDA, you do an IND package, many of you have done that, your IND enabling studies, and you try to prove that your drug is safe and non-toxic. But many of us, and I remember Kasper, and we had a preparation to this meeting, he said, well, actually in the future, we may have to take several drugs. You may have to take a drug during an early stage of disease and so on. So what about combination treatments? And maybe you should start, Kasper, if that's your vision.
Well, the neurodegenerative space is complex. Neurodegenerative diseases are complex, and I think the general consensus is that they are multifactorial. There's been two important successes, I think, in drug development in ALS very recently. They're for ultra-rare forms. One for SOD1, which is about 2% of the patient population. One for FUS patient, which is still a clinical trial ongoing, which is less than 1%. But there are people that are treated by those therapies that don't progress anymore, according to the clinicians that I've spoken to. And so that is not the average of the population that is being treated. And so for most people, there's still progression, but there's a significant delay in, or slowdown of that disease progression. In principle, we all think we need combinatorial therapies to really stop neurodegenerative diseases completely. I think we see that now back in the Alzheimer's space. I think it's incredibly exciting that a new Alzheimer therapy was approved last week. It slows disease progression by about 30%, so there's a lot still to do. But it gives us a foundation on which we can add. And I do believe that there's dominant drivers in a specific patient that have a bigger impact in that patient versus another patient, and that a certain patient would benefit therefore more for a certain combination, and another patient would benefit more from another combination. We see that in oncology. We can now do genome testing of plasma or take a biopsy of tumors, and we see that there are several different mutations in a specific tumor, and an oncologist will therefore use several different anti-cancer drugs to treat a certain patient. I've experienced that. I'm sure you've all experienced that as well, unfortunately, with friends and family members. My vision is that we're going to get the same for neurodegenerative diseases, and that we have put down the first stakes in the ground with some effective therapies that we can build on.
So, Lisa, I know you've been in, as you say, a business leader as well. How do you see that? CNS is already a difficult organ to treat. Do you see, obviously Kasper is making a very good point, but do you see that evolving? How would it evolve? Does it depend on surrogate biomarkers, or what do you think we need?
I can't say I have a great answer on how it evolves. I think what you said was very elegant. I'll take a very simple approach. We now have lecanemab out there. We have donanemab coming. With donanemab, they treated until plaque was reduced a certain amount, and then they stopped, and the patients were okay for a while. So I sit back and I go, is that a time when another therapy would come in? we have to build on what's before us. And right now there are pretty limited, although very exciting new options in Alzheimer's disease. Will we ever get to the point of completely personalized therapy? I have no idea. I feel like the quagmire of CNS diseases, to myself as a business person, you have microglial activation, astrocytes are being paid attention to, inflammation is always a problem there. We're focused on cellular regulation. Oh, and then there's all the plaque, right? You listen to all these companies working all of these spaces, and to parse them out, this is a long journey. So we're hoping right now, let's improve the ability of these patients first, and we'll get to what the combinations look like or the more unique treatments as we go forward.
So Rob, I know you're from several companies and you were working with new technologies all the time, right? You've had data even from worms, combinations of genes that play roles. How do you see that now after, you have such an experience and probably a great perspective. So what do you think about combination therapies? How do you deploy them?
Yeah, I think deploying them, really is going to become a de facto approach in neurodegenerative disease clinical trials. We already see this in ALS, where patients are on standard of care therapies and your therapeutics are tested on top of them. I think as we move forward in Alzheimer's, patients will be on these standard of care therapies that have shown to be disease modifying and will need to show additional benefits. I think that does become a complication and challenge for the field, but it does allow us a chance to explore novel biology and novel ways for targeting these diseases through different molecular approaches, again, to target different pathological processes.
Right, so I know Charlie and I have had some conversations over the last week or so, and I know you had some things that you think are important. So what would you like to ask the panel about in terms of new drugs?
Yeah, so I want to follow up on what Lisa said earlier, right? I think we have seen, perhaps made a little dent into treating Alzheimer's disease with the plaque lowering. Just think about how maybe what are the next catalysts that you think that you're expecting that could potentially help advance the field further, whether it's Alzheimer's disease, whether it's ALS, or the other kind of brain disease areas.
Yeah, what are the catalysts, you think? Targets, methodologies?
Who are you asking?
Kasper, do you want to start?
I'm happy to. Okay, I'll go first. I think a big catalyst actually just happened with Chris Wiebacher mentioned it, the approval of tofersen in ALS was approved through the the accelerated approval on a surrogate endpoint, a biomarker called neurofilament. It is that type of approval process that really accelerates drug development. And so now we have a surrogate endpoint, at least for that SOD1 population, but we hope that can be expanded to other ALS populations that will speed up our therapeutic development and maybe also make it more cost-effective, touching to this other problem that was addressed today about having affordability of drugs. We need to bring the development cost of therapies down. But if we can keep ALS patients alive, we're going to be in that same era as MS was before, and MS was also really catalyzed using a surrogate endpoint biomarker in drug development. And so I think that is the type of progress that we really need to see. We have a really good targets now to target the disease, but we need to speed up development through a precision approach.
Yeah, I fully agree with Kasper. I think the transformation in the field for ALS with neurofilament and predicting efficacy downstream is really going to enable us to design faster and more informative clinical trials. There's also new biomarkers that are emerging, things that we can measure at home, movement, speech, at-home functional assessments that are also going to be transformative for the field and really change the way we do clinical trials and enable us to accelerate development of these therapies.
I concur with these gentlemen. I think biomarkers is going to it's the lowest hanging fruit that I see. So before we were looking at, okay, you have to do a lumbar puncture, you have to do a CSF draw, PET imaging. You have to find people who want to do it, people who are skilled to do it, neurologists who are happy to spend the time doing it, or patients who live near imaging centers. What a game changer it will be when you can do blood-based diagnostics or follow trials in between with a less cumbersome set of ways of monitoring the disease. We were so excited at CTAD 2 years ago, C2N, now we have this ratio of A-beta 42 to 40, and that was valuable. It was a very small step, but now there'll be other things that allow us to get to a broader group of patients a lot more easily. You know, the ASI guys did a great job. I think if I remember this from their presentation, they had maybe 15% Latina patients, 5% African American in their trial. I'm trying to remember the demographics. I am sure lots of you out there know how incredibly hard it is to get diversity of patient populations. We want people who have the disease in our studies. Well, it's really hard to get them. And that changes what we know about disease. So if we can lower some of these barriers to more easily access patients or let patients access the trials, I suspect we will learn a great deal about these potential treatments. So the biomarkers could be a great way of getting there.
I think you got a great answer here. I think what the catalysts are, I think all three of you echo that, is that what speeds up drug development here is that we're going to get access to that personalized data faster. And I love what you said about blood. People don't think of brain disease as having any blood relationship, but actually, for those of us who worked in neuroinflammation or inflammation for years, worked on metabolic disorders for years, worked on lipid diseases where there are shared genetics, there's probably going to be a little bit of an eye-opening series in which the catalyst could be the human data that then informs us how to do clinical trials more effectively, frankly.
And to add to that point, Ole, like the TDP-43 discovery that I just mentioned, if we think about what TDP-43 represents, it's more than 90% of ALS patients, but it's also half of the frontotemporal dementia population. It is patients with late, a newly classified form of dementia. It's a subpopulation of Alzheimer patients, patients with PLS, and there are a number of other indications. If we can identify those patients through a biomarker, and we would be able to treat based on a biomarker, I think that would be a true catalyst also. And we see that play out in the oncology space again, to use that as an example, that framework actually already exists. If you have a specific tumor, you do a blood test, there is a medication for a specific therapy, then that therapy can be prescribed for you. I think that is the future of neuro. I know from a payer's perspective, we have to find a solution to that. And obviously, somehow we have to prove that a therapy works for all these diseases so that we can actually substantiate that. But if we can break down these barriers of these very dogmatic classifications between diseases, like we know ALS-FTD is the ALS-FTD spectrum. But you know, neurodegeneration as a whole is kind of a spectrum, and there are different disease mechanisms that play in multiple of these diseases that we should be able to identify with biomarkers and then bring the right therapy to the right patient.
I think that's an excellent perspective. It certainly also matches what I believe is going to happen with multi-omic data, very high levels. So again, from Charlie's perspective, you know, if you read the news, you see what drugs are approved and what targets have been addressed. Many of those targets were discovered 20 years ago, and it shows you how much effort goes into it. So I know you have other questions about what is the next, or how would you like to frame the question?
Right. So I think, you know, we talk about biomarkers, we talk about, right, everyone knows about A-beta, everyone knows about tau, and obviously, you know, Kasper has mentioned, you know, several kind of targets already. But, you know, maybe this is a question for all the panelists, like what are the next kind of exciting targets that you think, you know, that could change the field? I know it's a difficult one, and you know, perhaps it's related to your company, but we'd love to you guys to delve deeper into that.
Any billion-dollar bets here?
I love that question because it's obvious the answer is our targets. No, all joking aside, like, and I mentioned it at the beginning, you know, the missplicing in ALS is a product of the use of human patient-derived stem cell models and postmortem tissue analysis. That's a very recent discovery. And the therapies that we are bringing now forward and that are in the clinic are basically based on that discovery. And those, I think, are the best targets at this moment for this disease. It is different, perhaps in the future. As I said, if we can prevent disease, that's a different approach. But yeah, we wouldn't be doing what we would be doing if we wouldn't like our own targets.
Yeah, I'm particularly excited about our increased understanding around some of the core cell biology that occurs across the indications, particularly, and Ole knows this from some of my past, where there's evidence for lipid dysregulation across a wide variety of different diseases, and this can manifest in different ways to promote synuclein pathology or A-beta pathology, as we see with ApoE as one of the largest risk factors for Alzheimer's disease. So I think this area of understanding the core cellular dysregulation and the underlying processes that drive that are really going to turn up some novel targets that can be meaningful across these different diseases.
And I would just echo where we're working is at that cellular level, looking at alpha-synucleinopathies, A-beta oligomers, what happens when you can prevent their binding and displace them. So I'm quite confident as we go forward, we may be able to find things even beyond sigma-2 as a target that's going to be helpful in disease. We're in the clinic with 5 trials, so when you get to that point, it's like the efficacy signal, the safety profile, then you go back to really interesting science based on knowing you can get to patients and be safe and provide some type of a therapeutic offering for them. I think that's how it works. For myself, where we are, that's how it works. You're in the clinic, and then you're going to go back and invest in further science based on what we've learned there.
Yeah, I think what we shouldn't forget is that there have been a lot of failures in the past, but the last 10 years have given us a genetic revolution, a stem cell revolution, and a technology revolution. And all of these things are just now translating into clinical programs. And so the targets that are brought forward are much better than the targets that we had in the past, because we really now understand disease. And we have a really good way of measuring target engagement, mechanism of action, efficacy with biomarkers, and we tie that up with functional readouts. And so, we're going to see the first fruit of that product, and actually we're seeing it already, and it looks good. And so I'm really excited about where we are in this space, and I personally think for ALS, and I hope that I'm right, that we can turn this into a livable disease in 10 years, and if we can, then I'm also convinced that that will translate to other indications like frontotemporal dementia, and that would be a really a success, I think, and we'll be sitting here together and being very happy about what we've accomplished all together.
Charlie, how does that sound?
That's great. Perhaps if we just go maybe a little further, just to kind of think all the things that we mentioned, right, earlier. Like, how would you say a good way to improve that probability of success, right, into the next stage? You mentioned there's biomarkers, there's the humanized the genomic data, but I guess from like a practical standpoint, like how should we think about it? I guess from the investment perspective, like what are the things we should look for can help us figure out, what are the good data sets, what could be paradigm-changing treatment?
That is a great question for investors. I mean, Anne Klobinski said it best. She talked about the long cycle getting to patients. So for investors, my God, you have to have an appetite to wait a long time to find how these new mechanisms are working. I do believe, as has been said here, there will be multiple approaches going forward, and the willingness to take bets on them is key. You mentioned at the start of this, there's, there are genomic approaches, targeted genomic approaches, RNAi approaches, small molecules, large molecules, many modalities and different targets. And 10 years ago, I bet it was a lot easier for investors because they just had to find the best A-beta company with the people that had the most degrees and partnerships with the big companies. I don't think that's true at all anymore. I think you're going to have to look at a lot of innovative scientific companies and decide where you're going to place bets. If you look at the literature, the amount of funding for A-beta-oriented companies 10 years ago, 90% of the funding, the NIA would say this as well, had to do with A-beta-oriented plaque removal. Today, that number has dropped dramatically. 90% of the efforts getting funded, maybe not that much, are in a whole different set of approaches. So for investors, it's pretty hard because you're trying to find things that are additive to what's already been out there, and most of it's not precedented. I mean, for the great success of Leqembi, which is huge, we're like lost in the woods, you know, we're just at the beginning. In terms of truly understanding, there is no singular target here, so there's not gonna be a singular solution either. So it's about having an appetite to look broadly at a variety of mechanisms.
So maybe I can moderate those comments. So I think what I see is that if I look at it from the investor, a public investor, even public company investor, what I see on the innovation side is that analysts and investors are actually asking better questions than they did 15 years ago. And that what you hear from Kasper, Rob, and Lisa is that they expect to have more creative solutions. How do you create the subpopulation of patients? What's your surrogate biomarker? How you drive the process. So not just the standard chemistry to hit your target, right? But they're asking tough questions, and if they're going to get funding, they have to show some innovation to speed up the process, and maybe not make it 8 years, but maybe 6 years, right? And so I think it's a great question you have, but I think in terms of what I see, as we heard about Alzheimer's disease, I remember when the EMA said we maybe use surrogate biomarkers. That was 2016. So now we're expecting this wave, right? So I think we hear from all the panelists that they are expecting to use these tools and speed up development, I hope, or be more successful maybe, probability of success.
I think there's one thing that I think has really helped in the Alzheimer field. That is the understanding, and this is not my field, so Lisa can correct me if I'm not saying it right. The understanding on how to run trials effectively. I think at a helm, it was not completely done right, so it was a kind of a murky result that led to a murky approval. But lecanemab, very clean trial, very clear results. Donanemab, very clean trial, very clear results. We're learning that, and we're learning understanding our patients, again, through biomarkers, but also many other things. We're learning on what patients to enroll in the trial, at what state of disease. Yeah. Really getting clear answers with low variability means we need to have homogeneous patient population. And then we can extrapolate that and bring it to a broader patient population. I think that has really helped the AD field. And I think that is really something that investors are also looking to. Understanding your patient, running smart trials, keeping it cost effective so that it actually can be run in a small company, and then getting it to registration.
Now you move next door from AD to DLB, dementia with Lewy bodies, right? It's the biggest dementia you've never heard of. You can't get physicians to easily recognize the conditions. These patients spend 1.5 years getting diagnosed, 3 doctors, 7 visits, and the whole time their condition's going like this. I listened to the CEO of Vigil and I thought, oh my God, the families of the patients with these conditions, what it takes to get a proper diagnosis. So Alzheimer's looks like it's, you know, the golden-haired child because there's clarity around what that looks like. So there's just so much more work to be done in related dementias.
What do you think, Robert?
Yeah, I would say for me there's 3 pillars for increasing the probability of success in neurodegenerative disease and other disease to bring therapeutics forward. It's about designing the right trial, so innovative designs that allow us to test the hypothesis in question. Second pillar of selecting the right patient population. Are we going after the right patients, and how do we select those patients? Then the third part is really, for me, is understanding and a thorough understanding of the drug properties that we're testing. Do we have markers of target engagement? Do we know we're getting the right dose? Are we engaging the right pathway? Are we having an effect on a disease-relevant endpoint? Having a thorough understanding of this really de-risks moving into more expensive trials and improves our probability of success.
Then you have the rating scales, right? If everybody does right what you just said, Rob, you're still sitting there with ADAS-Cog 11, 12, 15, or 25. Can you count backwards by 7? Can you draw a clock? Not to diminish these things because they're standardized tools, but I feel like on that end, I hope too the progress you articulated, we see in capturing true improvements in patients.
Yeah,
it's
absolutely.
Pretty variable.
Absolutely, Lisa. Sorry for interrupting.
Yeah, no.
Yeah, I think as we get more precise endpoints that move away from these subjective rating scales, I mean, ALS-FRS is still the only approvable endpoint for full registration in ALS.
And survival.
And survival.
Which is a horrible endpoint because it means you put patients in a placebo group and wait until they are no longer on the face of the earth.
But I think as we advance more of these innovative endpoints, starting with NFL, moving into at-home measures, movement, sleep measures, particularly in Parkinson's disease, looking at other aspects of the disease, such as REM sleep behavior disorder, I think can really be transformative.
And the FDA is receptive to that. The tofersen approval, the SOD1 therapy was based on the totality of the data. They actually did not meet the primary endpoint in the phase 3 study. But by following patients for a long period of time, by linking up target engagement with efficacy biomarkers, neurofilament in this case, the FDA has agreed that there is enough evidence to support an accelerated approval. And so I think that the regulatory landscape is also really developing, and I think that's really good for neurodegenerative disease companies.
That's a great point.
So we hear, and I think this is a great contribution by the panel here, that the innovation gap is also, and I always think of this because I actually was a member of one of the panels at the FDA and CBER, that they are innovating too. They're listening to you, and in particular in the United States, I would say, it's one of the advantages is this kind of confrontation, but also innovative style. So the FDA is developing, and they're developing with companies and drug developers. So we have a few seconds left. I have to tell you, we have a technology gap. I'm not getting the questions on this panel, and I was told to waive it or something, but I hope if you have follow-up questions, we're all available by email on the participation. But I'd like to thank my co-moderator and all the panelists for their contribution. Thank you very much.